Yes, so I'll have a pretty broad overview of 3 different projects that all deal with the progression of neurodegenerative disease. And so we'll start off with modeling disease progression throughout the brain. And as you'll see, it's quite similar to how infectious disease will spread throughout, for example, a country. And then I'll introduce modeling how that disease has an impact on the neural activity, so the functioning of the brain, which is of course important. And then lastly, we'll look at a very interesting new avenue where it's actually becoming clear that neural activity itself can impact the disease progression. The neural activity, of course, works on, is based off networks, and the spreading is also based off networks. Right? So this first part is together with Dani and Michael Henderson. So we're interested in neurodegenerative diseases. What are they? They're diseases like Alzheimer's, Parkinson's, ALS, frontotemporal dementia, a ton of different diseases. And they have many things in common, and we'll only think about or consider part of it, which is that all of these diseases have toxic protein aggregates. So when you look at their brains, we'll find the cells have these clumps of proteins. Right? And what's interesting is that each disease seems to have their own protein that is associated with that particular disease. And these proteins, right, the cell isn't producing them to be mean to itself. They're normally healthy functioning proteins, but they have some modification happen to them where they become toxic and they start to aggregate. Right? And I'm interested in particular in a kind of proteins that are said to behave in a prion-like way. And tau from Alzheimer's disease, which is the protein in Alzheimer's disease, is prion-like. Alpha-synuclein in Parkinson's is prion-like, so prion-like aggregation is an important aspect of these diseases. What is prion-like aggregation? So imagine that you have the healthy protein and then an abnormal or modified toxic protein. Every once in a while, the healthy protein will be converted into a toxic protein, but it's quite rare. But the toxic protein can start to aggregate and form these big lumps. What's special about prion-like aggregation is that the misfolded protein can recruit these healthy proteins and turn them into toxic ones. So there's this autocatalytic conversion. And what's also interesting is that these prion-like proteins are known to travel down axons, essentially travel through a neural network and transfer from one cell to another. So this is quickly beginning to look like some kind of infectious disease. You have some toxic proteins, they turn healthy ones into toxic ones, and they spread from cell to cell. And this all culminates in what is called the prion-like hypothesis, which is a hypothesis that a number of these diseases start like this. You assume that for whatever reason, we normally don't know, there is some kind of misfolded protein already in some region. Then they start to self-replicate, they grow in number, they spread throughout the brain, they start to self-replicate, and so forth. Now, we know that these proteins behave like this from laboratory, experiments in laboratory settings. In vivo, in humans, you can't just open up brains and look at what's going on. So that's why mathematical modeling might be useful, especially in trying to test this hypothesis. So these models are network-based, where diffusion MRI and tractography is used to create these networks where nodes are brain regions and edges are axonal connections. And then we have these networks that are usually from about 100 nodes to maybe 1,000. And then we can model the spreading with different models on top of this network.
So this is actual structural connection.
Yeah, real.
Those images generally are functional. On the right, they show that you lights up,
Yeah.
This study is really the fact that there are some actions that goes from one place to
Yeah, these are real physical connections. Yeah, yeah.
the MRI means
Oh yeah.
fMRI is functional.
Yeah, I can explain that real quick. So diffusion MRI, well, it's a kind of imaging where they, I don't know how, but they track how water diffuses throughout the brain. And it turns out the water diffuses more quickly along axonal connections. And so that is used, so we get essentially like a vector field of like, where things are going. And then there's some kind of like parcellation on top of that creates the networks. Yeah. And so if the prime-like hypothesis is true, we should be able to fit these models to empirical data and get good fits. And there are tons of studies doing exactly that. And so there's a growing consensus that these computational models support the prime-like hypothesis. But there are some unresolved questions that I want to focus on, which is that even though you can guess, where the disease spreads first, some regions are just flat out much more susceptible to disease than others. We're not quite sure why. Another thing is that the pathology doesn't just increase, it can also decrease. So what is going on there? Why is it decreasing? Not really sure. So this section, I'm going to try and formulate a model to tackle these questions. Fit this computational model to data, protein data, and then correlate those optimal modeling parameters with genes to try and weave out what kind of biological processes are related to the regional vulnerability and decreasing pathology. So over to the data. It's really magnificent data. It's from Michael Henderson at the Faneuil Institute in Michigan. And essentially, mice are injected with this misfolded protein in a particular brain region. In this case, it's called the putamen. And then the mice are euthanized. They're killed. They take brain slices. They have then 2D slices. They stain them so they can see what proteins there are. And then you estimate the number or estimate the portion of these slices that have pathology. So that's an indirect measure of how much pathology there is in a region. And so here we have 1 month post-injection, 3 months, 6 months. Here's a particular region where we see an increase and a decrease. We don't have the actual brain network or structural connectivity of these mice. So we're using a publicly available reference network, which is average over mice from the Alain Brain Institute. And we have 448 regions. It's a directed, weighted graph. And I want to bring this home that for each brain region of the 448, we have one time series. We have 8 time points, multiple samples per time point. Many of the time points are in the beginning. And you see there's one thing to note is, of course, the mice have to be killed, right? They have to be euthanized. And so 1 dot, that's 1 mice, right? You don't follow 1 mice throughout time. Can't do that.
So these are experimental data.
Yeah, exactly. Yeah.
Of like, is it PTI?
It's actually that I'm not sure.
But it is like coarse regions of
yeah.
forming.
Yeah, exactly. So with edges being axonal connections. So yeah. And so I'll introduce 3 different models of increasing complexity. And then we're going to try and test out which of these models provides the best fit. And then we're going to move on from there. So from this weighted directed adjacency matrix, first of all, what we're going to use as a basis is an out-degree Laplacian. And so the Laplacian is defined such that this differential equation gives you the flow of a species, protein species, whatever, throughout the network in a way that conserves the mass. So this is the simplest model possible. So here we imagine that U is the protein concentration in each region. We really only have one parameter because L comes from the data, comes from the network. And that's the time constant of the spread. All models need initial conditions. We're lucky because we know exactly what they are in this case. There. We assume them to be 0 everywhere apart from the region where it's been injected. But the issue here is that you don't have that self-replication that we saw. And so the simplest way to introduce that is to introduce this nonlinear term here, which is often called Fisher-KPP style dynamics, where we have this global parameter that determines the time constant of the growth of the protein in each region. And we have a carrying capacity that is regional, so a carrying capacity for each region. How we understand this equation is, of course, if you just remove the spreading, what this tells you is if u is 0, you have no growth. If u is non-zero, you grow towards beta I. But of course, that is not exactly what will happen when you have both terms there. So it doesn't, that's one of the issues, is that this is a very you just had the one brain.
Recording in progress.
Okay. So this is kind of the simplest descriptive model I can think of. But as I said, in many cases, the pathology decreases. So what's the simplest thing we can do to make it decrease? We simply make this carrying capacity change with time, make it decrease in time. So now we say that we have the original carrying capacity here, so that this starts out at beta I, and it will decrease by an amount of this that is specific to each region. And then all of this happens, the decrease happens with a time scale that is the same across all regions. Okay? So now we have 2 times n, 2 times 448 parameters, 2 parameters per region, and then we have 1, 2, 3 global parameters. How we're going to fit this model, so we're going to use a Bayesian approach. So I'll just describe that very, very briefly. So imagine you have a vector of modeling parameters. You have some data, and you want to infer what the modeling parameters are. In the Bayesian approach, you want to find the posterior, which is the probability of the modeling parameters after having observed your data. Bayes' theorem, which is here, tells us that we need 2 things to find that posterior. We need the prior, which is the probability of the modeling parameters without having observed the data. And we need the likelihood, which is the good old normal likelihood, the probability of observing the data given your model. And so multiplying those together, you get the posterior. And we're finding that posterior with MCMC sampling method. So the priors, we don't have any particular reason to pick one prior over the other. We know roughly by playing around with the equations that they should be on the order of 1. And why not use the normal distribution? When we're going to constrain all the parameters to be positive. And so we simply just say, look, this is normal around 0 with a standard deviation 0.1, so forth. We're also inferring what the seeding concentration is, because we really don't have any biophysical units here.
Yeah, this is my problem. So there is no physical units.
Yeah.
So there is, okay, why 0.1 and not?
It's no particular reason. I mean, we don't have any better reason to use something else. If you do play around with the differential equations, though, and you put in gamma equals 100, alpha equals 10,000, and rho equals 10 to the minus 6, you're going to get crazy looking things that wouldn't be meaningful.
Yes, is informed by the fact that the equations are behave in a way that is.
Yeah. It's also based off of previous studies that have used these approaches, and they've used normal distributions with good fits, essentially. And so the likelihood, how do you compute that? We sample the parameters, solve the equations. We get a prediction U of I of what the level of pathology is in each region. And then we simply say that the data is normally distributed around that prediction. So not looking at the mean of the data, we're literally using all of the data points. Okay. And that normal distribution has a variance that we've assumed to be constant, just for simplicity's sake. And so I'll show you some of the priors and posteriors with the decay model, with kind of the full decay model. So here we have the prior, yellow line. We run 4 instances of the sampling to make sure that we approach the same posterior distribution. And we get a nice unimodal looking posterior for this diffusion constant. Same for the decay and for the aggregation. This is just to show you that they don't do anything super funky. The carrying capacity, here's an example from region 42, whatever region that is, and then also decay. Some of the regions don't really update that much from the prior. It typically happens in regions where you have very little pathology at all, because then you can't really, how are you going to tell what the carrying capacity is in a region where you don't have any pathology, right? So now we're going to compare models. First, it's informative to just look at kind of an observed versus predicted plot. It's on log-log, not because I'm trying to, hide anything. It's because of a lot of the values are very small, and so we'll look weird on a normal plot. And so we take all of the time points, all of the regions, and the average in each time point in each region. Gives us 448 times 8 data points that we're plotting with the prediction of the mode of the posterior, so the most likely parameter value. What we find is that the diffusion somehow catches some kind of ordering correctly, but it's way off by orders of magnitude. The aggregation looks relatively okay, and the decay as well. But the big difference is here, where the decay does better, and these are the later time points. That'll be clear here. So here we're looking at the actual data in blue with the mean and standard deviation of the data in a particular region, where we have sampled parameters from the posterior, simulated the model, and then we can compare and see, what it looks like.
So they have basically, this is for region 76, they have this data for all regions, the 448.
Yeah, there's quite a lot of data. Yeah. And what we see here is that the aggregation model is kind of awkwardly trying to go between these two here. But at least to the human eye, this looks a bit more comfortable, where you see this rise and fall in the decay. Same here for another region. And here's another region where there is no decay. So that does happen, that you don't see any decay. And luckily, the decay model doesn't do anything crazy like doing something like that. Yeah. But we can't just stare at plots all day and say, oh, the model is better. It's better to maybe use some model selection metrics. So here we've compared all the models with the archaic information criterion, the widely applicable information criterion, and the Bayesian information criterion. These all in different ways rewards the model for having high likelihood and penalizes for having complexity. That is a lot of parameters. Okay? The more negative it is, the better it is. And consistently, the decay does better. So, okay, fine. We're going to go through with the decay model. So what was the original research questions we were interested in? It was, what causes this regional vulnerability? Why are there decreases in pathology? And now we have these two parameters that our thesis is, in some sense, captures the vulnerability, the beta, the carrying capacity, and the decay parameter. So we then have this gene expression data set for a subset of the brain regions, not all of them, unfortunately. Quite a lot of genes. We correlate these genes with the modeling parameters. And what we find is that the genes that have to do with cellular metabolism are significant more often than you would think according to by chance. And here we are looking at the R value. So if it's negative, it's negatively correlated. If it's positive, it's positively correlated of the carrying capacity and decay. Each dot is one gene, so it's 20,000 dots here. The larger they are, the more significant is the correlation, the lower the p-value. And what we see is that the carrying capacity is negatively correlated with metabolism genes, because we've painted the metabolism genes in blue. And we find this inversion, where essentially there's a negative correlation with metabolism to vulnerability. And for the decay, there's a positive. This is very recent. We produced these plots on Friday, so we're still looking, So this is just like a taste. So yeah. So there's more to come in the future. But that's the first part. So I've shown you these computational models. They can, in some sense, depends how convinced you are, capture the spatio-temporal spread. Transport, aggregation, and decay seem to be necessary. All these three things. We had this result with the cellular metabolism that we're still working on. But as I mentioned, this is a descriptive model, right? There's no mechanism here. The parameters don't correspond to anything in particular. And I would like to build a model more from the bottom up, maybe connecting the neuronal scale, and then maybe epidemiological models might help out there. So over to the next section. So this was part of my PhD, which I did together with Alain, Goriely, and Bick, and another collaborator from the Amsterdam Cancer Research Center. So he's a neurologist, also working with Alzheimer's patients. As you can guess, we're going to switch over to Alzheimer's for this section. But kind of the general theory still applies. So obviously, you don't think about protein aggregates when you think about dementia. You think about cognitive decline, memory loss, so forth, memory dysfunction, really. And that presumably comes about from the death of neurons and from synaptic dysfunction. Yeah. The main theory is, of course, that misfolded proteins are to blame for this. And so we'll be talking more about Alzheimer's disease and delve into that a little bit more. And then we're going to try and connect this model of prion-like spreading with neural activity models. So in Alzheimer's, there's two proteins, so it's more complicated, unfortunately. We have amyloid protein that spreads around, amyloid beta, and tau. Amyloid beta starts very early on in the disease, and tau starts later. Amyloid, it's kind of not entirely clear where it's beginning. That's why it's a bit diffused here. Tau almost always start in the entorhinal cortex, which is this region here. Now, there's an interesting shift in the way we're thinking about Alzheimer's that has occurred in the last 20 years, where some researchers are suggesting that there's two phases to the symptoms in Alzheimer's. And if we follow the blue line, which is supposed to represent neurophysiological activity, neural activity, the hypothesis is that it's increasing first years before symptoms come, and then decreases. Okay. And this is how you will start having cognitive symptoms. We know for sure this happens in mice models, for sure. Does it happen in humans? Not so clear. But there's some evidence in EEG and MEG, which I'll just very briefly introduce in a toy schematic. So essentially in EEG, you put electrodes on the scalp, and MEG, you have this larger machine around you that measure changes in the electromagnetic field on the scalp. What people have found for nearly over a century now, actually, I think it was first discovered in 1910 maybe, is that you find these oscillations. They'd be called brainwaves or neural oscillations, and there's a big field dedicated to studying these. The bread and butter is, of course, take a Fourier transform of your signal and look at what kind of frequencies there are, correlations between frequencies and so forth. Different frequencies, there's evidence that different frequencies are associated with different cognitive processes. Now I'm going to make it very simple. I'm just going to care about biomarkers, in the sense I'm just going to care about what we know about neural oscillations that change during Alzheimer's. So when you're during rest, you have typically, especially in the back of your brain, you have frequencies, neural oscillations at 10 hertz. And this peak that is often observed in the periodogram is called alpha peak. Right? And there's some evidence that there's more alpha power in early stage Alzheimer's this is very difficult to study because how do you find early stage Alzheimer's? How do you find people that are going to have Alzheimer's, but you don't know it yet, right? But it's very much established that there's a decrease in alpha power and that this alpha peak is slowing down in frequencies. Even so much that some hospitals use the slowing of the alpha peak in diagnosing patients with Alzheimer's. So it's not just fantasy. What exactly it means, what it does to the alpha power, I don't know. But that's on the clinical level. Okay, what is happening with the molecules and all that, the proteins? So there's this great review paper by Harris that is very long and complicated, but where they put forth this hypothesis, which I've called the excitation inhibition hypothesis. And I know this is a lot of information at once, but stay with me. So what they're saying is that amyloid beta affects different neurons differently. So amyloid beta will activate excitatory neurons, that is neurons that excite other neurons, and it will deactivate neurons that inhibit other neurons. This has been established in vitro and in mice. So tau only affects excitatory neurons, and it affects axonal integrity. This is, a lot of information, but what's nice is that we can use a mathematical model to test this hypothesis. And how we're going to do that? We are going to use a diffusion MRI tractography derived empirical structural connectivity brain network. That was a lot of jargon. A brain network of physical connections. We're going to model the spreading of amyloid, of tau. But then there's also this whole field called whole brain modeling that is trying to model EEG and MEG signals. It's called whole brain modeling. In these models, invariably, there's some kind of like so-called neural mass model, where in each region you have excitatory and inhibitory neurons. They affect each other. Classical motif for creating oscillations. I'm not going to go through the models. The idea is then that you have, one variable for excitatory neurons, one variable for inhibitory neurons that is evolving on each node in the network. Then we're simply just going to have these proteins, introduce them into these equations, and then have them change them throughout time. And then we're going to test, does this hypothesis produce the changes that we know are happening in Alzheimer's? So imagine this is a brain region. We have a parameter associated with the excitatory neurons, inhibitory neurons, and then we implement our hypothesis. And then we ask, do we see the early hyperactivity, late hypoactivity, and do we see the slowing? So I'll just show you the simulations for the protein progression, where we have a network of around 90 brain regions. But we're showing the results that are averaged over different lobes of the brain. And it's just to show you that we have splitting of amyloid first and then tau later, which is known to be the case. Amyloid comes first. Here we have the evolution of the parameters of the neurons. So this is parameters associated with excitability for excitatory neurons and excitability for inhibitory neurons. And we see this two phases, essentially. So now it's a matter of pick points throughout time, simulate the real neural activity, the simulated neural activity, and then see what happens. These neural models, we had some random parameters, so we repeated the simulations multiple times. That's why you're seeing standard deviations here. This is showing the alpha power, so how much alpha oscillations you have. We see an increase and a decrease. And furthermore, we see this slowing down of the frequency.
What is the time scale here? So because you don't have an actual time scale in the early years yet.
Yeah.
So what is 30 what?
Oh yeah, so you can change the parameters and pick yourself, so to speak. Yeah. Because it's like in physical units, again, the same models.
It would be 30 years or 30 days.
Yeah. So essentially, how you're thinking is that this is where symptoms start. Takes about 10 years. Don't count me on it, but I think it takes like 5 to 10 years from symptoms come before the individual might die from Alzheimer's. That should happen around here. And then people don't really know when amyloid beta comes because it's incredibly difficult to ascertain. But some people throw out numbers to say, 10 to even 30 years, you might start having amyloid before. So that's kind of the only way we look at the time scale of the disease progression. Yeah. And that's really the little I wanted to say about this project. It's very brief. Interesting thing here is that there's this hypothesis on the cellular level that is, that people from biologists have come up with, not clinicians, right? Biologists who are studying proteins, and then we link that with a mathematical model to symptoms observed by clinicians, and we find that they are consistent and work well together. We have a follow-up study where we're fitting the spreading model with real PET brain, so that's protein data, imaging data for proteins, and fMRI, which is a notion which measures the notion of neural activity. And now, lastly, I want to also briefly introduce another project where you're completing the loop. We talk about disease progression, linking it to neural activity. But fascinatingly, the neural activity also may affect the protein spreading, which creates this quite complicated dynamical system. We have two dynamical systems on the same network operating at different scales that are both communicating with each other. So there's actually kind of like two projects here. There's one that was part of my PhD and a new one that is together with Dani as well. So very briefly, why do we even think that neural activity may impact protein spreading? There's this paper from Karen Duff's lab where, as before has been demonstrated many times, if you have in a laboratory setting neurons here, axonal connections, and you put a protein here, it will show up in the other population because it's traveling through the axon and being transferred. But what they found was that if you artificially make the one neuron fire more, you get more protein in the other population. And so we said, okay, well, first of all, just from a mathematical point of view, this is very exciting. Let's try and come up with a model that combines these two processes. And now, because these results are really on the neural level, the networks I'm talking about, is we're not really fitting the data. We're just thinking of them as neuronal networks, not brain region networks. So first we have the protein spreading. And now we're using a slightly more complicated model than what we had before, which has been of the heterodimer model. You will notice it's quite similar to an SIS type model, where we now have in each region a healthy protein and a toxic protein, right? We have the spreading designated by the Laplacian of the network. We have production of the protein, decay, because proteins are produced and they are destroyed naturally, degraded. And then we have a conversion term from healthy to toxic. Toxic protein is not produced by the cell, or so it's assumed. But we have a decay term. They're still being degraded. It's a very simple model. It, in general, has two dynamical regimes, a healthy or disease-free. Yeah.
So the K0 is just a low, is natural production.
Yeah, exactly. So if you imagine that you just looking at one cell, no conversion, no transport, it will grow towards K0 over K1. Essentially, we have two dynamical regimes here, a disease-free state where you don't see production of toxic protein, or if you change the parameters in a certain way, you get production of toxic protein. The neural activity, we're kind of cheating because we're not really using a neural model. We're using phase oscillators because those are very well understood theoretically, and we know they are in some way related to neurons. So imagine that each neuron has a phase associated with it, an angle, and we're just going to say the quicker its phase is accelerating, the more active it is. So quick is active, slow is not active. And then each neuron is going to have a natural frequency or a natural fire rate. And then they're connected sinusoidally to other neurons in the network. I've forgotten to put in, usually you have the weighted adjacency matrix terms here, which I forgot. But so this just looks like a complete graph. But this essentially has two regimes as well. This coupling strength is either very large, or let's say it's very large or small. If it's very large, you get phase locking. That's just a jargon for synchrony. They're now synchronized, the neurons. If you have low coupling strength, they're drifting, which is jargon for asynchrony. And so what's the simplest way we can couple these? We can make the natural frequency of each neuron decrease by the local concentration of toxic protein. So the more toxic protein it is, it will fire less. And then we're going to say, well, what do we mean by activity? As I said, we're going to say that that is equal to the frequency of the oscillators. And that turns out that's just the time derivative of the phases. And then we're going to scale the transport of the protein by that activity. And I'm just going to show you one theoretical result from this, and then one numerical simulation. And so essentially, for the theoretical one, we're just going to work with two neurons. Just because we're mathematicians and we want to look at the two neurons. So it's kind of ridiculous to say that at the Network Institute. But yeah, anyway, here we go. So we say, okay, well, this very simple, the most simple configuration of two neurons. Simply put, we can understand the dynamics quite well by just considering two parameters: the toxic clearance, that's how quickly the neuron is removing toxic proteins. You want that to be high, right? Because then you're removing as much toxic protein as possible. And then neural activity coupling. And so here we have a bifurcation diagram that I'll walk you through real quick. Y-axis, we have the protein clearance. X-axis, we have the coupling. So this is the parameters, right? And each part here designates a different parameter regime where you have different long-term behavior of the system. So there's this red line here, which turned out to be, in this model, surprisingly simple. It's just a line. To the right of this line, the neurons synchronize. Simple as that. When they're to the left, they are drifting. They're not synchronized. What's interesting, however, is this line here, which is the critical clearance, which we could find analytically, which show you where you go from being in the healthy regime to in the toxic regime. But what's interesting here is as you become less and less synchronous, you increase the space of the toxic regime. Why is that interesting from the disease point of view? Because you could imagine if we shamelessly generalize this to a network, and you imagine you're here, you're healthy, you don't have any toxic proteins in your brain, then theoretically, right, you could just change the neural activity, keep everything in the brain exactly the same, no changes in anything of the protein dynamics, and then you could be pushed into a toxic regime. We tested this out on a small random ER graph of 10 nodes, effectively just seeing if we saw the same thing. So here we're looking at the stationary solution, the long-term level of toxic protein in the different neurons. So here we see, here they're asynchronized. We see different levels of toxic protein. And this is the toxic clearance. Around 1.7, we start seeing disease. Whereas when they're synchronized, it happens further down. So this is worse if you want to stay healthy, so to speak. So for me, the interesting takeaway from the previous, which is the only part I showed you really. The interesting takeaway from that project is that it seems like the neural activity itself can initiate the disease. In healthy people even, not only in Alzheimer's disease, in this region, the medial temporal lobe, you will see tau accumulation. So in healthy adults that don't have any symptoms, they actually get this accumulation of tau. And that's also, if you remember, the region where Alzheimer's begins. So the difference really between Alzheimer's and healthy aging is that the tau spreads out. Yeah. And medial temporal lobe is where you find the hippocampus, the amygdala, so things that have to do with memory. So maybe that's why age-related, you get age-related symptoms to memory loss. So what we wanted to ask is, well, could it be then that the neural activity is responsible for this accumulation in the medial temporal lobe? If it's so that, theoretically, it can initiate disease. So this is also a work in progress, and I'll just show you one figure from this. So what we wanted to do is to introduce some data. So now we're back on the whole brain scale, where we're now using imaging data. So imagine you have it's better to go here. So we have this structural connectivity brain network. We can extract this 18F FDG PET imaging. What is that? That measures how much glucose is being consumed in brain regions. Neurons that are very active need a lot of energy, so it's an indirect measure of neural activity. And so we're going to use this to have like a rough measure of how active each brain region is. And then we're going to say, look, in this simple model, we're just going to say, in each brain region, we have a very small but existent production of these toxic proteins, but very low. And then we're going to say that they can be transferred between regions, but that is affected by the neural activity. And what's really cool then is that we find that it is exactly the medial temporal lobe that gets the most accumulation. And we also find that this is true regardless of whether or not you have Alzheimer's, whether or not you're late stage Alzheimer's disease, early stage Alzheimer's disease, or whatnot. So in this part, we looked at this mathematical model of activity-dependent protein spreading, and we show that it can at least provide a mechanism for how tau starts to accumulate in the medial temporal lobe. So very briefly, just to remind you, we've gone through a lot here. So just to remind you what we've done in the first part, we looked at protein spreading itself and fitted it to empirical data in mice data. And we find that we can find reasonably good fits. In part 2, we coupled such a model with neural activity and did that so we could do hypothesis testing of how the proteins affect neural activity, providing evidence for this idea that Alzheimer's has 2 phases. And then we looked at a model where we completed the loop, and we found that neural activity can initiate the disease spreading. And we showed that with some empirical data that might be the case for tau in healthy adults and Alzheimer's disease. So that's it for me.
Question. Yes, please.
Yeah.
How do you assign the natural initially is should depend on individual threshold of
Yes, so for the numerical simulations, we simply put them to be random. We just distribute them randomly. But you're entirely correct. We're also working now on kind of like redoing that model, but with more realistic neural models, quadratic integrated and fire neurons that have this threshold. And then in those models, those are assumed to be also have some distribution, a Lorentzian distribution. Yeah. So, yeah.
My question is, I'm still a bit puzzled by, so in other models, there is never this any time scale in a sense. And this is what makes me a little bit uneasy in a way, because you have in the more in the other models I can understand that you have one parameter that is the beta, and the scale you can adjust. So I would say it's a matter of deciding a time scale and then measuring the two parameters of that time scale and so on and so forth.
Yeah.
But here you have 4 or 5 parameters.
Like in the last one.
In the last one.
Let me find it. Yeah.
I expect at a certain point things should break down if some of these K0. So if K0 is 0 is different, if K1 or K2 are completely. So how you are guided into that? So this is there any information that can be used here or not? So these are the action rate. So I assume that you must have at least a few guesses on what is the rate of generation of those toxic prions, etc. So there is no experiment that says, okay, I inject the prions.
So they do that in vitro, so in containers, and there I know for a fact they do have aggregation constants and more complicated ones, even because there's in that case when people are modeling prions evolving in a container, they're looking at they're modeling specifically healthy protein, misfolded protein, but 2 aggregate, 3 aggregate, 4 aggregate, 5 aggregate, blah blah blah. Yeah. So here, one thing that I will say though is that we do find in the models consistently across different groups. I haven't put in the row here that would scale the Laplacian, but that one, when you're modeling on the brain scale, is consistently a lot smaller than the aggregation constant. But whether or not that fits with biological experiments where you actually look at how quickly do they transport and how quickly does the aggregates form, which I'm sure you should be able to get. I'm not sure if you will.
Just kind of ideas of the time scale separations that you have in the different processes. I imagine that there is something like that. I think this scaling by diffusion constant is okay, but then it's also the relation of the other parameter. The other question that I have that is technical is in the part 1, where you have the Bayesian approach to get the posterior of the parameters.
Yeah, you wanna here. Yeah.
Here, am I correct that you have a beta I? So you have 400. So you are doing something with 440 parameters because beta I and the I change for each region.
It's even more because the K is also 450, so around 900 parameters. So
it's 900 parameters.
Yeah. And
you are able to run the MCMC with 900 parameters.
Yeah.
Wow.
It takes 2 days. So one of the reasons maybe why that is working is because, there's tons of data.
Because this is a 900, at least my experience with MCMC, you have a 900-dimensional phase space. You have to sample, you have to do your little moves in this. It should take forever.
So we are using what is called Hamiltonian Markov chain Monte Carlo, and that's I don't know much about it. It's state of the art, blah blah blah. But so maybe that's something to do with it. I'm not sure. Another thing though is that the pathology. I don't have a figure of that at hand, but the pathology, because it's when they inject the proteins, it's on one side of the brain because they don't inject. Because you basically have mirrors of regions, right? So injecting one of them. So one side has a lot of pathology, the other does not so much. So most of the data will just be a line because there will be no pathology.
It might be that, with your very well-defined minimum. It's a little, generally MCMC, when you are ready with 4 or 5 parameters, you have it's quite. I understand that your equation is easy to in a sense to simulate, and for you,
that's true.
It's very quick the simulation.
Yeah.
I was surprised. Okay, I need to look at the technical details in a sense.
I will say though that one thing that has bothered me for a long time is that this likelihood is really not sensible, and that because that goes on to your point, because the it shouldn't be normally distributed because now there's a probability that the data is negative, and the data can't be negative because it's percentages. So that's annoying. So I've tried out different ways of having a more realistic likelihood function, but then the Markov chain, the sampling method just takes, like, it's inconceivable. Like I can't. So I'm using this because I'm forced by how slow the sampling method is. So it's working quite stable now with these priors, but yeah. So
yeah,
that's one comment at least.
This is more technical. We will get into that. So please.
So I have one that also Giovanni Petri from Parkinson, London has a question.
Great.
That I'm trying to embody. Can you predict secondary effects of protein transfer, as in high activity nodes accumulate more proteins reducing activity, which then results in other nodes trying to start to do more, et cetera, If so, maybe in the model you might see the net diffusion vector go converting.
Yeah, so if I understood it correctly, first of all, I will say that our collaborator Willem de Haan, he does have a paper that's not related to this model, but where they look at fitting a model to, like a spreading model to empirical data, and they find that hubs do have more protein, but having to do with it, I don't remember exactly, but I think that might be something I can send later. One thing I will say so do I have the plots here? I don't think so. But so one thing is that I will say, we thought we were going to see more complicated phenomena with these two time scales, but we did not for a particular reason. And that was that imagine that you have, equal amount of toxic protein, and some are more active than others. The one that is the least active will get the most protein. But if the protein is deactivating that neuron, that will just make that even less active. And so if you imagine a high active, low active, this one will decrease quicker. So through time, they will never cross. And so when you have that the protein is deactivating the neurons, I don't think that will ever happen like in an inversion. But if you have positive, then you might see an inversion, maybe some more interesting things.
Got it.
Yeah.
There's like a bunch of questions that I have, so I'll try to keep them down, but I think they're really nice. I really like the talk.
Thank you.
Maybe just to start with Alex's question at the very beginning about these are structural networks, not functional connectivity. I'm just curious, do you think it's feasible to imagine some of this spreading happening in a sense via functional connectivity, in like in a way where these neurons are synapsing more because there's some sort of functional relationship to each other, and so there is this adaptive network process as well as the dynamics by now.
I definitely think so, and there are people looking into that specifically. So there's a researcher in Munich called Nikolai Franzmeier, and he's looking at this in particular. So he started out using functional connectivity to investigate the spread of these proteins, because he told me he had the functional data and didn't have the structural data. So he's like, okay, I'll just use this. And then it turned out that it worked quite well. And it's can't say with 100% certainty because it's a long time since I looked into that paper. But I believe they looked at doing it with structural connectivity, estimating the spread with structural connectivity, then functional connectivity, and then a mix, and then finding that the most parsimonious best model is also with functional connectivity. Yeah.
I guess they're just related to that. So my own interest in some of these questions on neuroscience and network science come from like reconstructing functional connectivity when we have time series.
Yeah.
And it struck me in, I think, the middle project that when you have these models of excitatory and inhibitory dynamics that are producing some activity, especially when you're trying to emulate like neurodegenerative diseases or pre-clinical stages, like the actual ability to reconstruct some network from some of these time series, because the time series are kind of messed up by inhibition and excitation out of whack.
Yeah.
It's actually maybe more or less reconstructible per se in those periods. So it just it made me think about like this, because you touch a lot of this data, but if there's something that is some signature of, let's say, Alzheimer's in time series readouts of some of these or time series data in patients, if you think that there may be some word that we as like networks people should have when even trying to think about network reconstruction in these kinds of great sense. I don't know if that question made sense at all, but it seems like there's something that's pretty, I mean, the dynamics are quite different in a clinical stage versus preclinical versus healthy.
No, I think that makes a lot of sense, because you can then leverage different, especially if you have, and you will have, I'm not sure, but if you have the same individual throughout time, then that might be really, really interesting from that reconstruction, because then you essentially, assuming that not the axonal connections doesn't just like completely disappear, which they won't, then I can see there being a lot of data to leverage from that.
Curious to talk more, but also
Yeah. Okay, well then let's thank you. For those of us here, you have us. There's